My dear learner, a very warm welcome to all of you. Myself Ankita, and in this session we will discuss about the biomolecules. Now let us discuss about polysaccharides. The word polysaccharide consists of two parts. First is poly and other is saccharide. Poly means many and saccharide, which is from the Greek origin, means sugar. The acid insoluble pellet also has polysaccharides, or you can say carbohydrate, as another class of macromolecules. The polysaccharides are long chains of sugars or the monosaccharides. The monosaccharide means one sugar unit. Mono means one, saccharide means sugar. These are the simplest type of carbohydrate. The monosaccharides having either a free aldehyde group, aldehyde means CHO group, or a ketone, that is CO group. So the monosaccharides are divided based upon the number of carbon atoms in a chain. That means how many carbon atoms are present in a chain. So let us discuss about the type of monosaccharides. First is triose. The triose are monosaccharides having chains of 3 carbon, that is, first is aldehyde group, second carbon having one H group and one OH group, the next third carbon, that is CH2OH group, that is alcoholic group. Next is tetrose. Tetrose means 4 carbon in a chain, but basically there is aldehyde group which is present in the first carbon. You can say these sugars are aldose sugar. Why aldose? Because aldehyde group is present on the above of the chain or on the first carbon. Second carbon, which is having H group and OH group. H and OH can be on the left and the right side, but their position is always on the side chains. Next carbon again carries one H and OH. Then next is CH2OH group. Example is erythrose. I will also like to describe here that alpha and beta forms and L and D forms of the sugars. In the L form, that is known as levorotatory. In the D form, that is known as dextrorotatory. In the L form, OH group is on the left hand side of the carbon chain, but in the dextrorotatory, the OH group is on the right hand side. That means you can say that the L group or the L form of the chain, that is carrying OH group on the left side, and D form which is carrying the OH group on the right hand side. Next example is pentose. Pentose having a chain of 5 carbon atoms. Their examples include ribose and deoxyribose. In the ribose, there are 5 carbon atoms in a chain. First carbon carrying the aldehyde group, second again H group and OH group, third is also similar to second, then fourth, then in the last fifth carbon carrying the alcoholic group, that is CH2OH. These 5 carbon atoms also having ring forms. Ring form for 5 carbon is known as pentose or furan ring, but in case of 6 carbon atom, these have different names. Next example is hexose. Hexose having a chain of 6 carbon atoms. Their chain is having 6 carbon atoms and aldehyde group again on the first position. Then second carbon carrying OH and H groups. Then third again H and OH groups, likewise in the similar way. Here I am discussing about the examples include glucose, fructose and galactose. Let us come to the structure of glucose first. The structure here is glucose, which is basically the D-glucose and also an aldose sugar. Now here D is for the dextrorotatory. The OH group is on the right hand side. Which OH group you are watching here? For the carbon chain. The carbon chain is also in the ring form. Now I am describing you the alpha and beta forms. These are also the further classified structures of the chain or the ring form. In the alpha form, you can say in the carbon which is having number first carbon, the OH group is below the ring. If the OH group is below the ring, you will say this as alpha D glucose. D is for the OH group which is on the right side of the chain. But in ring form, alpha represent the carbon which is carrying the first carbon and OH group is below the ring. This is alpha. But in case of beta, the OH group is on the upper side for the first carbon. So, alpha and beta are different just from the position of the OH group. In alpha form, OH group is below the ring and in beta, OH group is above the ring. Next structure is for the fructose. Fructose is a ketose sugar. Why ketose sugar? Because here no aldehyde group present, there is present ketone group, that is C double bond O group. You can say in this diagram, the carbon which is at the second position carries a ketone group, that is C double bond O. In the ring form, this is ring which is known as furan ring, furan for fructose, and it is a member of 6 carbon atoms but carries a ketonic group. The ketone group is represented by the O on the upper side. Further, the fructose is divided into two groups, alpha group and the beta. As I already have told you that alpha form where the OH group is on the first carbon is below the ring and in beta form it is above the ring. Here in fructose, OH in alpha position is below the ring but in the upper position there is CH2O group, but in comparison to glucose there is H, because in fructose the last carbon carrying the CH2OH group. Next example is for the galactose. These all are the actually hexose sugar. Their structure varies just because of the H and the OH or the aldehyde or the ketone groups. The galactose having structure very similar to glucose. The difference is only at the carbon number 4. In glucose, OH group is in the right side. As you can see in this diagram, I am showing you the structure of glucose and galactose. See, at the fourth carbon, the OH group in glucose is below the ring, in the ring form, but in chain form it is in the right hand side. But in case of galactose, the OH group in the fourth position is on the left hand side and in ring form it is in the above side of the or the upper side of the ring. These both structures are very similar. The difference is only at the carbon number 4 which carries the OH group. Next is I am telling you about the haptose. Haptose having a chain of 7 carbon. The word hapt means 7. Example include pseudohaptulose. The very interesting thing about monosaccharides is that at least having 2 OH groups bonded to their carbon backbone and one aldehyde or ketone group. Based on these aldehyde and ketone, they are aldose sugar or ketose sugar. Because of this, these monosaccharides tend to dissolve easily in water. When dissolved in water, the chain form gets converted into ring form. Usually the glucose and the fructose, these are in the chain form, but when dissolved in water, these get converted into ring form. Now tell you the disaccharides. The disaccharides are formed of 2 sugar monomers. An important example of sugar in nature is sucrose, which is formed of glucose and fructose. The individual monosaccharides are linked by glycosidic bond. This glycosidic bond is formed by dehydration. The process normally removes the water molecule. This bond is formed between 2 carbon atoms of 2 adjacent monosaccharides. You can say that one monosaccharide is separate entity and other monosaccharide is another separate entity, both are joined by a bond. This bond is known as glycosidic bond, which will be formed by the removal of water. Let us take an example of disaccharide lactose. Lactose is a sugar or milk sugar with one glucose and one galactose. Here is a picture which is depicting you the structure of lactose. In the lactose, one monosaccharide is galactose and other is glucose. As I already have told you that in galactose, the OH group at the fourth carbon differs from the glucose. In this diagram, the carbon carrying the OH group on the upper side in galactose, and in the glucose, the OH group on the fourth carbon is on the below. By the removal of a water molecule, the bond will be formed between glucose and galactose, and this linkage is known as 1,4 linkage. Why 1,4 linkage? The first position for galactose, carbon number 1, and fourth position for the glucose at the carbon number 4. By the removal of water, the bond formed is HOH and the product formed is lactose plus a molecule of water. This is disaccharide and is created from the monosaccharide. Table sugar that we use commonly in our homes is sucrose. This is extracted from sugarcane and sugar beets. Sucrose is formed by glucose and fructose. Glucose is one monosaccharide and fructose is other monosaccharide. Glucose is an aldose sugar and fructose is an ketose sugar. Again by the removal of water, how the water will be removed? The water will be removed from the first position OH of glucose and H from the fructose. Then this will form a water molecule and the bond formed is glycosidic. This bond is 1,2 linkage. Why 1,2 linkage? Because the carbon number 1 is from the glucose and carbon number 2 which is from the fructose. The bond formed between both is 1,2 glycosidic linkage. But in case of lactose it was 1,4 linkage. These are disaccharides. These are created by the monosaccharide itself. But how? By the formation of glycosidic bonds. In a polysaccharide, the monosaccharide are also linked by glycosidic bond. They are threads, literally a cotton thread, containing different monosaccharide as building blocks. Polysaccharides are straight, branched, these are chains of monosaccharides. The polysaccharides formed from same monosaccharide, these are known as homopolymers. You can say that the polysaccharides which are formed from the similar kind of monosaccharides are known as homopolymers, whereas which are formed from the different monosaccharides are known as heteropolymers. For example, cellulose. Cellulose is widely present in the plant cell wall. This is a polymeric polysaccharide. That means it is formed of several monomers of which unit? Basically the glucose. So, glucose is a monosaccharide which is formed in chain form in the cellulose structure. So, cellular which is formed of same type of monosaccharide. So, it is known as homopolymer. Here is a structure which is representing the structure, basic unit of the cellulose. The glucose rings are arranged in a chain form or you can say in a linear chain. And the bond formed is between the first and the fourth carbon. The first carbon OH group is taken and then the fourth carbon again the OH. By the removal of water molecule, glycosidic bond will form. Like this, several glucose molecules will arranged in a chain form and the structure formed is known as branching or further linear chains ally in a chain form, and this is known as the homopolymer polysaccharide of cellulose. Cellulose does not contain complex helices and hence cannot bind H2. This means cellulose structure is not a helix form, so it can't hold iodine. The plant cell walls are made of cellulose. Paper made from plant pulp and cotton fiber is also cellulosic. In cellulose, glucose chains are arranged parallel to one another and this form a sheet. This bonding arrangement where chains are stretched stabilize the chain in a tightly bundled pattern. So, this is the reason why they resist hydrolysis by most enzymes. This is also the main feature of the plant cell walls, having fiber sheets of cellulose which gives toughness, insolubility and resistant to weight loads and mechanical stress like strong winds against stems. Starch is a variant of this, but present as a storehouse of energy in plant tissue. So, in plants there are two types of homopolymers are formed, one is cellulose and other is starch. The plants which carries starch, the starch is divided into two categories, amylose and amylopectin. The difference between both is just a bonding pattern. In amylose, the bonding is alpha 1,4. Now you can say that the alpha form which is having OH group on the below side of the ring is bonded to other carbon which is from the different monosaccharide and the bond formed is first and the fourth carbon, that is 1,4 glycosidic bond. But in case of amylopectin, the bond formed 1,4, then other bond that is known as alpha 1,6. Now what is 1,6? First carbon carrying the OH group on the below side, that is alpha, and that is attached to the sixth carbon carrying the OH group. By the removal of water molecule, bond formed is 1,6 glycosidic bond. So, amylose and amylopectin are the basic unit of the starch homopolymers. Further classified products of starch are the helical secondary structure, that means starch form helical form. Now, as I already have told you that cellulose can't hold iodine molecules because that does not have helical portion, but in starch it can hold iodine molecules as starch can contain the helical portion and can hold iodine molecules. The starch iodine is blue in color. This is a test done for the presence of starch in food. Animals have another variant called glycogen. The glycogen, which is very similar to starch and cellular, just the difference is the large branching patterns. In the cellulose, chains form, sheet form, in the starch, helical form, but in glycogen, more and more branching and branching similar to amylose and amylopectin, alpha 1,4 and alpha 1,6. Here is a picture which is describing you the basic structural, or you can see the basic linear arrangement of chains and branch. In amylose just a chain, amylopectin more branching, in glycogen very compact branching, in cellulose sheet form. But these were the homopolymers. Why? Because they were formed the same glucose subunit. In a polysaccharide chain, say glycogen, the right end is called the reducing end and left end is called the non-reducing end. In animals, glycogen is a storage polysaccharide which is equivalent to starch in plants. Muscle and liver cells in animals have lots of storage in glycogen. At the time of decrease in blood glucose level, liver cells degrade glycogen and release glucose which enters the blood. So, that means glycogen is the major storage polysaccharide in animals. Insulin is a polymer of fructose. It has branches which are shown in the following diagram. The branching is very similar to the glucose subunit which were in the cellulose, starch and other glycogen. There are more complex polysaccharides in nature. They have role in building blocks, amino sugars, chemically modified sugars like glucosamine, N-acetyl-lactosamine. Exoskeleton of arthropods, for example, have complex polysaccharide called chitin. The arthropods whose exoskeleton, that means the outer skin which is formed of the chitin. This is the heteropolymer. Why? Because not only the glucose, several other groups are joined to the glucose polymer. So, that is why they are known as hetero. Hetero means different group other than the one. In chitin, there is side group containing nitrogenous base that is attached to the glucose unit. This polysaccharide aid in strengthening the external skeletons and other hard parts of the many animals like in crabs, earthworms, crustacean, insects, spiders and ticks. Their outer skeleton is formed of chitin. It also gives strength to the cell walls of the fungi. So, to summarize what we have discussed about polysaccharides, biomacromolecules, metabolites, further primary metabolites, secondary metabolites, their function, their role, their actual structure and also the necessary products also. Thank you.